Okay, so the next company is AlzeCure, and with me I have CEO Martin Jönsson. Welcome and take it away.
Thank you so much, Fredrik. Great being here. Thank you for being here. Martin Jönsson, CEO of AlzeCure. And AlzeCure, we are focusing on Alzheimer's disease and pain. We are a spin-out out of AstraZeneca. So when Astra left the CNS field, researchers from Astra founded AlzeCure. We are sponsored from the Swedish Alzheimer's Fund. If it wasn't for them, we wouldn't exist. And we still have a close collaboration with them. We are based at Karolinska Institute, and we were actually founded out there. And we only work with small molecules. And this has been a very conscious choice because we think that based on the big patient populations, we need to have oral drugs. We have the drug Alzstatin, which we are developing to prevent that you develop Alzheimer's disease. And then we have Neurology, which we are developing to improve learning and memory capability, but where we today also have data indicating potential disease-modifying abilities. And then we also have Painless, our pain platform, where we in certain projects actually are turning around the effect in Neurology, where we are upregulating signaling, and in Painless we are downregulating the signaling. And we are listed since 2018. Our business model is that we are a research and development company with the aim to out-license a couple of our projects to then drive the other project further. And this is the pipeline we are having with our 3 platforms. In Neurology, we have positive data in phase 1 with also target engagement data showing that it's safe and tolerable and activating centers in the brain which are key for depression and learning and memory capabilities. And here we are now preparing for phase 2. And then in Alzstatin, which is a so-called gamma secretase modulator, we have a new molecule that we developed last year. We have these internal capabilities to build molecules. And we are now choosing between 2 candidates to choose one to take the lead candidate into clinical trials. And in Painless, we are here having a project focusing on neuropathic pain. We have positive phase 2 data in chronic patients with neuropathic pain, and now preparing for the next phase 2. And then our second pain project focusing on osteoarthritis. We have developed our own TrkA-NAM molecule and are now preparing also this for phase 1. Our pipeline has been developed in close collaboration with leading experts such as Henrik Zetterberg, who is a world leader in Alzheimer's disease, as well as Bengt Winblad and Maria Eriksdotter. We have also recently had Jan Lundberg, who was head of research and development at Eli Lilly, who is standing behind the fantastic portfolio that Lilly today has. And Jan has invested in our company and also joined the board. So now we are working actively with Jan. In Alzheimer's, we have 2 platforms, Alzstatin and also Neurology. What's great with these programs are that they are actually validated by big pharma. So both Roche and also Eisai have similar projects that we actually can compare with. And it's also creating confidence in what we are focusing on. In Alzstatin, we are focusing on to diminish the buildup of toxic fragments in the brain and plaque, which is destroying the neurons. The process for developing these plaques and toxic fragments is that there's A beta 42 being cleaved out and then building up into plaque. What we are doing is that we are diminishing the production of A beta 42. We can do that with over 60%. It has been shown that in individuals who have a lower amyloid A beta 42 production of 20 to 30%, in these families, they do not develop Alzheimer's. So based on that, there should be something around amyloid. And what we are doing is not only diminishing A-beta 40 and 42, we're also increasing the production of the shorter forms, A-beta 37 and 38. And it has been shown both clinically and preclinically that these shorter fragments can be protective. What we are focusing on and have targeted is several different patient groups. We see it as a maintenance treatment in patients who have developed Alzheimer's. We also see it as an opportunity to combine it with antibodies. And then the ultimate thing is to have a preventive treatment. Finding the patients based on biomarkers, genetics, and also looking at amyloid beta. And the tests already exist today. We are preparing for phase 1, attractive profile. So why? Because already in this trial, we see that we can show not only safety and tolerability, but also how we can diminish the buildup out of these toxic forms of amyloid beta. And here we also plan to have a patient cohort to early generate attractive data. And finding and identifying the patients, we will use biomarker kits, which is already available today. In this field, we are trailing behind Roche. They've already had a successful phase 1 trial showing that they can diminish A-beta 42 and safe and tolerable. How do we differentiate against their molecule? We think we have a more potent molecule. Our second program, NeuroRestore, to improve neurotrophic signaling. Here we are preparing for phase 2. We are focusing on the neurotrophins, which are key for brain health and learning and memory capability, and that is BDNF and NGF. We know that individuals who have a lower levels of NGF have brain deterioration. We also know that individuals who are lower on BDNF also have a quicker disease development. And here we think that by increasing the signaling of these neurotrophins, we can improve learning and memory capabilities and also potentially brain health. What we are doing is that we are increasing the signaling of BDNF and NGF. And what we can say is that when we developed initially this program, we were focusing on improving memory and learning capabilities. But now we also have data that is showing that we are also, as it looks, have the ability to improve brain health, improving neuroproliferation, neuroprotection, increasing neural outgrowth, improving synaptic health, mitochondrial activity, and also anti-inflammation. So when we add ACD856 to the neurons and add A-beta 42, we see that we can protect them. So here we see a potential also for disease modification. But we have also shown that we, in preclinical models, can improve learning and memory capabilities, so the ability to learn, store, and remember. And we have shown this in preclinical model where we give old animals our compound. We give one dose, they perform a memory test, and then we test them after 10 days. Animals who have not received the compound more or less forget everything. Animals who receive ACD856 perform at the same level as the young animals who are at peak when it comes to learning and memory capabilities. What we have shown in our phase 1 is it's safe and tolerable. We have also shown that it has an excellent PK profile for once daily. We have also shown it goes over the blood-brain barrier. We have also shown that it activates centers in the brain which are key for learning and memory capabilities, but also for the treatment of depression. Eisai is also active in this space. They actually say that their compound can be a potential disease-modifying compound for neurodegenerative diseases in general. When we compare with the ASI, which we of course do, we can see that we have an excellent molecule which is well differentiated. We have a half-life suitable for once daily, which they don't have. We have data on brain health, improving potential brain health. They also have that. But what we also have data on is that we can improve learning and memory capability, but also for treating depression. We have now published on this, came out this fall, so you can read more about this. And then we of course also have our painless platforms. I will not go into detail in these projects today, but I can say that we have TrkA-NAM focusing on osteoarthritis, preparing for phase 1, over 300 million patients. And then we also have ACD440 improve for neuropathic pain, where we have positive phase 2 data in chronic patients. And this project is based on what received the Nobel Prize. And today at 1 o'clock, we are actually having a seminar around pain and pain disorders and also going through ACD440. So scan the code, join in and learn more about ACD440. And why AlzeCure? We are targeting areas of huge unmet medical need. We have a strong team that has created what you have seen in our pipeline and are continuing to develop it from idea into clinic. We also have first-in-class properties that we have developed that truly can be game changers. We also do parallel investments, so we are no one-trick pony. And the areas we are going for are multi-million dollar or billion-dollar opportunities. And we have also been able to show that we have developed from idea into a development company. So that's AlzeCure. So thank you for that.
Thank you, Martin, for a good presentation. You mentioned here that Eisai and Roche validate your drug candidates. Can you maybe just elaborate? Are they differentiated enough? Can they both coexist? Or what's your opinion on that?
They can both coexist, but they are also differentiated. If we start with Eisai and compare to Neurorestore is focusing on improving learning and memory capabilities. What Eisai have chosen is only to develop a molecule that potentially can improve brain health, but not to improve learning and memory capabilities. So based on that, we are well differentiated. If you look at the molecules, we also have a better half-life, etc. So I truly think that we can differentiate. When it comes to Roche, also there we can think that it's great that they are there because they are paving the way for us. We can see that with the interest from other pharma companies and also investors. And then synthesizing their molecules and compare to that gives us confidence.
Okay, so it could be a competitor to Roche, for example, that sees a modality that could work and
absolutely
want to have it as well.
Absolutely.
Okay, and you mentioned here that business development is an important part of your strategy, and you have said that from the recent rights issue, some of the funds would be to assist with that.
Absolutely.
What's the status right now?
That we are having many dialogues, and all of these dialogues take time, and that our strategy is to out-license 1 or 2 projects and to use these funds to drive the other projects forward. But it is long processes and it takes time.
Do you have a timeline, or just that as soon as possible, of course?
Both as soon as possible, but also to acceptable terms. But I mean, doing a deal, validating our business model, fueling the company, increasing the pace. That's great for both for the company and the projects, of course.
And you mentioned about the ACD856 that there are a lot of potential effects. It's anti-inflammatory, it's disease modifying, and so on. How can you utilize these findings in the clinical program? Can you can you prove it potentially all at once, or would it be separate trials or add-on trials later on, or how would that work?
Good question. When we look at what we are developing NeuroRestore for, it is to improve learning and memory capabilities, and that is really what the patients really want to have and see. Then, when we do these trials, we also want to look at potential disease-modifying abilities. As we see it today, we don't think that we will do separate trials for disease modification, because disease modification is also much more challenging and also demands much longer, trials. So we focusing on initially on improving learning and memory capabilities, and we think there's a huge potential there. We know that donepezil, their drug sold for more than 5 billion at peak sales, but only about 25 to 30% of the patient could endure the treatment, and their efficacy wasn't very big. If we develop a drug which don't have these side effects and also have a great efficacy, we think the market can be huge. And also, if we can also show that the compound can be used for treatment in depression, which there is science supporting. There has been publications in Nature around showing that the psychedelic compounds drive their antidepressant effect through the TrkB BDNF receptors. There's also been publications in Cell showing that ketamine and SSRI, also that their effect, as it looks, is driven through the BDNF TrkB receptor.
That's interesting. And you have two Alzheimer's projects in different stages of the disease. Can you just explain the kind of how they would be used in the patient journey? Could it be used at the same time or not at all?
We think they can be both be combined, and then initially often we find these patients too late, so they already have memory impairment, and their NeuroRestore is a great potential treatment. When it comes to alstatin, it can be used hopefully both in patients who have developed Alzheimer's, but the ultimate is that we identify the patients early. We know today with the new test that you potentially can identify the patients 8 to 15 years before they develop the disease. And with the initial additional data showing that impacting tau and impacting amyloid buildup and everything, that if you can lower the production of amyloid beta and then postpone this disease development, then we think that alstatin would be a perfect treatment.
Okay, and yes, so you mentioned in the presentation that there are actually tests today that could be used to identify patients early. Can you expand on that and how far away are they from being mainstream?
I mean, if you are in the US, you can actually go to clinics and almost to the pharmacy and take this test. So available they are. And if you go to KI and have the right connections, you can actually also get the test there. But there they are used for research basis. p-tau 217 is the best one. p-tau 180 was used before, but there are also other tau tests being developed. So they are there. There are several.
And what will be needed for them to be used in a clinical and much larger setting than in the research
stage?
I
mean, what's very much driven this is that there is beginning to be treatment for these patients, because in many times, how should I say, they haven't been used. Also to some extent because certain groups have been hesitant to identify the patients already early. So now with treatments coming, all the phase 3 trials that's there, I'm sure they will be used to a greater extent.
So maybe the incentive hasn't been there because there haven't been any treatments.
Yeah, both that, but also the technology has become so much better, and Swedish research groups have done a great job there. And I think that's also a very relevant question there, because many of the patients who were included in the so-called Alzheimer's trials before, they were not Alzheimer's patients.
Okay, that's not good.
That's not good, exactly. Yeah. So with this improved test, we are able to identify the patients and making sure we have the right patients. But then also, since we are learning so much more about genetics and everything, we also understand to a greater extent which Alzheimer's patients should be treated with what treatment.
Yes, and this could maybe improve the likelihood of approval for all assets in the field potentially.
Absolutely.
So maybe we have time for a final question. Yeah. And I was thinking about ACD 440. Yeah. What could kind of be the next stage in a phase 2B setting, for example? What type of trial, number of patients?
It would be a POC trial. There we are discussing different kind of trial. It can be peripheral neuropathic pain, but it can actually also be an orphan indications. There are so many orphan indication that you also could have, and based on that you could do smaller trial and come quicker to the market. And I actually feel that the interest for orphan indication has grown over the last 2 years, since also the big pharma companies have been under economic pressure. So to find cheaper trials and quicker ways to the market, we see that that there is more interest and also more talk and more questions about orphans.
And when will you land on this? Is it based on partner interest, or?
It's both based on partner interest, but to do an orphan, you also need to have a pre-IND meeting or a meeting with EMA, and you can do all of these things in parallel.
Thank you very much, Martin.
Thank you so much. Thank you.